In Lab 12, we're going to do with just a few clicks of a button what takes a lot longer to do with Anova by hand. We've talked about Anova by hand having 9 different calculations. Well, JASP is about to make our lives a lot easier. Let's take a look. First of all, make sure that you've downloaded the data set and then download the lab instructions as well, and I've done that. So here are the lab instructions. First, let's talk about what this study is all about. So mice are used in an experiment to test drugs that might prevent the advancement of Alzheimer's disease. Half of the mice are what we're going to call transgenic, meaning they've been genetically modified to have Alzheimer's disease. And the other half of the mice are what we're going to call the wild type, meaning they haven't been modified at all. And the mice are assigned to one of 4 drug treatment conditions, 4 different drugs that are all supposed to help with Alzheimer's disease. And the mice are taught to run a maze 2 different times. The first time they run the maze, it's what we're going to call a training day. It's the first time they've encountered the maze. And the number of errors the mice make are recorded. And then after some time, the mice are then asked to run the maze again, and we're going to call this the memory day, because hopefully they have some memory from the first time. And the number of errors that they make on the memory day is also recorded. So in this study, using inferential statistics, we're going to do a couple different things. First of all, you're going to determine whether there's statistically significant differences in the number of errors that are recorded on the training day, depending on Alzheimer's status. So Alzheimer's versus no Alzheimer's. Did they perform significantly different on the training day? You're going to do that same type of test, but this time we're going to look at the memory day. So did they perform differently depending on whether or not they had Alzheimer's on the memory day? Finally, we're going to determine whether there were statistically significant differences in the number of errors recorded depending on the drug treatment. All right, so hopefully you've uploaded Lab 12 data into JASP, and let's go through and follow all the directions. So I'm gonna pull up my JASP here. So here are the data, and the first thing we always have to do is make sure that our variables are classified correctly. So the Alzheimer's status, Alzheimer's disease status is nominal, that is correct. But the treatment is nominal. There's no order to the treatment. JASP just thought it was an ordinal variable, but it's not. And then the training day and the memory day are both scale variables. They are on a scale, a ratio scale of measurement. So here we go. Next thing we need to do is we need to put labels for the different variables that need them, for the nominal variables. And the schematic for these labels is at the end of your directions, but I remember them by heart, so I'm going to put 1 was Alzheimer's, and 2 was wild type. That means the mice that were not modified at all. Let me make sure those took. That did. All right. For the treatment, we had 4 different drug treatments. The first treatment was a tau drug. The second treatment was amyloid drug. The third was a tau amyloid combo. And the fourth drug was what we're going to just call a cognitive enhancer, or COG enhance is going to be my label for that. I'm just going to make sure they all took, and they did. Very good. So now we're all set. So what do we normally do at the very beginning of our analyses? We're going to do our descriptive statistics. We're going to start with our nominal variables, move them over. I'm going to select frequency tables, and I'm going to deselect the statistics because I cannot do a mean or a standard deviation when I'm talking about a nominal variable. It just doesn't work. So as I look at what we have here, it looks like we are half and half. Half the mice were in the Alzheimer's condition and half were not modified. And it looks like there's an even split between the 4 different drug treatments as well. That's the value of looking at a frequency table. We can see how our subjects shake out. Next for the descriptives is going to be our scale variables. Now we can look for means and standard deviations. So as I look at training day versus memory day, it looks like the average number of errors on training day was 14.725. And on the day when they came back and reran the same maze, the average number of errors was 9.925. So it looks like they did differently the second time around. They had less errors. They had memory for the maze. All right, now we're going to do two different types of descriptive analyses. I'm going to back up here to descriptives. And this time I'm going to compare the training day scores, that is the dependent variable, based on the Alzheimer's disease status. I'm going to move that over here. So what did this tell us? Let's take a look. Well, it looks like on the first day, the training day, the Alzheimer's mice had an average of 14 errors and the wild type had an average of 15 errors. Looks like it's basically the same, right? So it looks like there really wasn't a difference in their performance on that training day. That's interesting. All right, and now we're being asked to do the very same thing again, but this time we're going to put memory day in the variables box and Alzheimer's disease in the split box. So let's see what we have here. So this time, the Alzheimer's mice made an average of 12 errors and the wild type mice made an average of 7 errors. Okay, this is interesting. Now we have a difference. What is the difference? Well, the first time they ran the maze, it didn't involve any sort of memory. They were just running a maze. And the second time they ran the maze, memory was involved. All right. So as I look now, I want to make sure I've done everything. Before we get to our Anova, what we're going to do next is we're going to do some independent samples t-test, because you are asked to compare the training day scores based on the Alzheimer's disease status using an independent samples t-test. So I'm going to come up to t-test and click on independent samples, and I'm going to move the training day scores over to the variables and the Alzheimer's disease status to the grouping variable. I'll click on effect size. I'm going to click on descriptives too. All right, so now we've done an independent samples t-test we only had two groups, Alzheimer's versus wild type mice, right? And it looks like we have a t of negative 0.991. The p is 0.328. It's not significant. There's really not a difference on the training day between the Alzheimer's mice and the wild type mice. We have the means are here that we already talked about. Okay. Now we're being asked to do another independent samples t-test, and this time we're going to put memory in the variables box and Alzheimer's status in the grouping variable. I'll click on effect size and descriptives again. And now I come back and I see, okay, well, now we have a t of 8.33. If you can imagine the t distribution, you know where that 8 would be. The p is less than 0.001 and Cohen's d is 2.635. I'm not going to give away the store, but you think about this. What does this say about the difference in memory scores between Alzheimer's and wild type mice? All right, so we've done all that we can do to this point, but we do have 4 different drug trial groups, and so we can't do a t-test. I mean, we could do a bunch of t-tests, but we'd be inflating our experiment-wise error. We don't want to do that. So what I'm going to do is I'm going to do an Anova to see if there's a difference in the scores on the memory day. That seems to be the day that is different based on the treatment type, the drug type. So I'm going to go to Anova and then click on Anova again. And this time I'm going to put memory to the dependent variable. This is the thing I'm measuring to see if there's a difference. And what am I looking for a difference in? The different treatment groups, the drug types. There you go, the treatment groups go there. Now in your directions you're asked to do a couple of different things. You're asked to click on Descriptive Statistics, Estimates of Effect Size, and make sure eta squared is selected. On assumption checks, you're going to click on homogeneity tests. We want to make sure that our variances are equal amongst all of our groups. And finally, in case there's a significant result, we're going to click on post hoc tests. I'm going to move treatment to this box right here, And I'm going to make sure that Tukey is selected. So I think that is all that we need to do. I'm also going to select effect size. There's one other thing. All right. And now I'm going to close this out, and we have everything we need to be able to analyze this ANOVA. And you're going to be asked questions about this in your first worksheet. So as I go back up to my ANOVA, I'm going to look for a couple of different things. First thing I'm going to look for is the ANOVA. So here we go. We have some information about degrees of freedom, and we've got interesting information about the mean square, but we have an F of 0.624. It's pretty close to 1, isn't it? And what we've talked about, when an F score is balanced at 1, it means there's no treatment effect, really. And our eta squared is 0.049, so you can look at the chart for how to interpret eta square and think about that. So basically, we have a p-value of 0.604, so you're going to evaluate that depending on whether it's less than 0.05, where we would reject the null hypothesis, or greater than 0.05, where we would fail to reject the null hypothesis. Our descriptive statistics have the means here for the different drug groups. And as you can see, it really looks like they're about the same. I'm not seeing anything that's standing out. We did an assumption check to make sure that our variances were equal, and they were indeed equal. I know this because the p-value is not significant. And this is the case where you want to see a non-significant p-value. That means there's really no difference in the variances. We're good. We did post hoc tests, but we don't have to even look at these, because when we had a non-significant result, we're not going to even look at the post hoc test. All right, that is it for now. Make sure you come over here and you export your results. And you can do that. It helps me greatly if you actually submit it as an HTML file. Okay, I've said enough. I'll see you at the next video.